Good afternoon here at Ars Electronica Home Delivery. My name is Sandra, and today I want to show you something very special from our Ars Electronica labs. We've seen parts of the material lab already, of the second body lab, and now I'm going to take you on a short tour around something that's very special in this area. It's actually quite nice when you come here from the second body lab, where you learn all about artificial limbs, artificially created skin, and much more. Now we're going to take a look on the inside. So the prostheses are more on the outside, and what we are going to look now is called organoids. And this has something to do, as the name already suggests, with our organs. But we have to make a slight distinction from the very beginning. We are not talking about organs as you can find them in our bodies right now. So not a real heart or a liver or a brain. No, that's why they're called organoids. So they are sort of organs, but we can do a lot with them. They're especially important for science, for research. They might even change the way how we treat certain illnesses. But before we're going to take a look at that, I'm going to tell you how this came to be, how the organoids came into existence. It was not that long ago that the two scientists, Jürgen Knoblich from Germany and Madeline Lancaster from the US, had the idea to come up with a way to do more than just create fiber and matter and cells in petri dishes. They thought, why can we not do something that's more three-dimensional? So to actually research it better and to get a more realistic picture of life. They've been working on the Austrian Academy of Sciences for a few years, and just in 2017 they came up with this idea and actually had a medicinal breakthrough, you could almost say. So they created an artificial brain, and not just a one-layer slice of a miniature brain. No, they created almost, I have to say under parentheses, a whole brain. How did they do that? Well, how do our brains develop when we are in the womb, when we are waiting to be born, when we are embryos? Our stem cells get the message delivered by certain proteins what they should become. And some of them become brain cells. We know how this process works now, and we can simulate it. That's exactly what those two scientists did. They took stem cells and fed them the correct proteins for them to grow naturally into a miniature brain. Also, we have to get one step back again, because how did they get the stem cells? Stem cells are not that easy to come by, because they are normally only found in the marrow of our spine or in the umbilical cord of embryos. And both things are not ready at hand when you need them. It's very expensive to get it. And so that's why they needed an alternative for this to work, for them to create brains on a relatively cheap scale. So they went back to the year 2012 to another scientist from China who had another breakthrough. And this time it was really groundbreaking. So the name of the scientist, I always have to look him up, Jinja Yamanaka, he came up with the idea of how to regrow stem cells from old skin and blood cells. So he didn't need the original sources of umbilical cords or spinal marrow. No, he took old cells, dead cells almost, they were still a little bit alive, but not connected to anything on the human body anymore, and repurposed them, made them evolve backwards into stem cells, which then again can be used again to create other cells. They can become skin cells or even become brain cells. And that's what they used here. So they took those stem cells, relatively cheap to come by, as I said, and fed them the correct proteins to develop. On the first few days in the dishes, you cannot really see that much. So we're gonna skip this a little bit. You have if you come here again at our museum, you can see how it's developed, how the proteins are given to the stem cells for them to create new matter, so to say, and to develop. But if you look at the petri dish here, there's nothing visible for the bare eye, at least. we have to move on a little bit more to say day 20. Here you can see a little tiny blotch. It's not much, but it is visible, hopefully also on the video. Otherwise you can see it on the screen what this tiny blotch is composed of. So after 20 days, the first neurons are developed. So not just stem cells anymore, they found their purpose in life, so to say, and became other things. From the stem cells, they became neurons, they started doing tasks, and they tried to connect to each other. And of course, this goes on as long as they have food, as long as they have proteins. And up until day 60, you can see a little bit bigger of a blotch, still quite tiny. But what you see here is this structure that we have here. So it's starting to get a little bit bigger, almost like this. So the size here, you would say, for my fingers is like this. So it's really a miniature brain, but with lots of important functions that we can actually observe. But don't be worried too much. It's not like this brain can suddenly have its own consciousness and start asking questions or anything. It is limited, and it is also very chaotic. Because in a normal embryo, we would have a normal development of the brain's sections. The neurons would know how to go to a certain place to connect with other neurons to, for example, form the cerebral cortex. Those cells don't know that, so they're quite jumbled up. They are messy, but they still have certain functions they can do, and they can grow up to a certain point, up to a year, approximately. Only the outer cells, though, because the inner cells start to die off. Why? Because this brain, of course, is just one single part, one single organoid, without a connection to a blood system, for example, to a blood circulation. And without blood, our brains wouldn't work, and neither does this one. So this actually shows us how far science has already come to recreate life, but to a limited extent. So like I said, this cannot be transplanted into anyone soon. Hopefully no one will need a brain transplant soon. But this is not the idea that this study is focused on. What we want to do with this little brain is actually do some research on it. Normally we have, for example, lab rats or mice for this kind of stuff when we want to know certain processes in the brain, how they work, how they can react to certain electrical impulses, for example, certain shocks, certain messages, certain scents. And it works up to a certain degree. But if you've had a look at the brain of a mouse, it's quite different than from our own. We can do a lot of research on it, and it has been done before. There's even a statue in Russia, in Novosibirsk, depicting what great contribution to science mice brains were, you could say. The statue is knitting a DNA strand, which I think is quite interesting. But here, this is, so to say, the next step to get away from mice and other animal testing to safe testing on something that does not have a consciousness. So this one can be taken to, for example, research the problems humans have with epilepsy, for example, or autism, Parkinson. Like all the illnesses we know come from the brain or certain damaged areas. Or even if you have a tumor, you can actually take a look at how we can make cells grow again to repair the damaged area. So that's the path this research is taking, to get away from animal testing and also to find a safe way to make us better understand certain illnesses of the brain. Of course, those organoids can not only become brain cells. If you want to, you can also create other matter, for example, heart cells. Thinking of heart transplants maybe in some near or farther future, we don't know how fast it's going to be, but this one has already been accepted worldwide, almost, you could say, by researchers to find an alternative for animal testing. Hopefully this will be made mandatory. I hope, that's my personal hope, that less animals will be tested in the process for scientific research, but it's definitely a good start, and we can do a lot with this little brain. Imagine like a few more years of research, what else will come of this. cheap method, safe method of research for everyone. Yeah, that's basically it for today. I hope you enjoyed this. Like I said, have a look at it when you come here at our museum. There is so much more to know about our organs and what we can do with them, even if they are artificially created. And I wish you a nice afternoon with this message, with this hopeful message that it's going to be better in the future for science. Thank you and goodbye.